5:35, so in the interest of time, we'll get started. Hi guys, welcome to the first IR Innovation Program webinar. My name is Raj Khalsa. I'm part of the resident fellow section of SIR. If you have any questions at all, I have my email down at the very bottom of the slide here, so please feel free to email me if you guys want to get involved with the innovation program or RFS or anything like that. I'm also part of the vascular disease service line, so if you have a specific interest and want to do some projects on vascular disease, we can definitely get you involved. So basically this presentation is quite short. It's about five minutes, and it's an introduction on this new program that we have. Today is the first webinar. We're very happy to have Dr. Arepally and Dr. Murphy here with us talking on an introduction to medical innovation and interventional radiology. And I basically want to start with this statement here from the American Medical Association. It's the Principle of Medical Ethics section from their statement in 1971, and basically says physicians should strive continually to improve medical knowledge and skill and should make available to their patients and colleagues the benefits of professional attendance. Basically, this was written in the 1970s, and at that time there wasn't a lot of medical innovation, and this really applied to the day-to-day practice of medicine. Since then, there's been a huge explosion in medical technology and innovation, and this now, this definition has a very different meaning than what it had before. When you're prescribing medications mainly, very few sort of instruments at that time. Now we really need to think about what that means in the context of medical innovation, device development, and we really need to be involved in that process altogether. This is a really good article that came out a few years ago. It's Physician Industry Cooperation in the Medical Device Industry. So this has been studied, and what basically they found was that physician involvement in medical device development is actually quite important. And not only is it important, it adds value to the actual device itself. It adds value in terms of a higher success rate of that device being patented, being marketed, becoming a successful product, and for subsequent inventive activity. And the best example of this is Dr. Palma's patent for the balloon expandable stent in 1985. And if you look at this graph from a paper that I'll show you shortly, there was a huge explosion in medical devices and interventional radiology based on that one sort of seminal patent that was huge for the field. So that paper, the physician industry paper, it also has this really interesting slide that says orthopedic surgery right now is on top. And radiology, interventional radiology is sort of middle to lower on the, it's down here somewhere, another one, basically down here, in amongst all fields. And it's really interesting because if you look at the history of medical device development and sort of the relationship with the industry, orthopedic surgery has really mastered that, and that's the AO-Synthes model that Dr. Murphy had kind of taught me about before. And it's really an extremely successful model, and what it is AO is a nonprofit organization that focuses on research in orthopedics, and Dupuis-Synthes, or Synthes at the time, was a for-profit organization that was born from the AO. And they've had a really fantastic symbiotic relationship since basically the late 1950s, early 1960s. And they come together every year. They train over 500,000 orthopedic surgeons every year, and their three fundamental sort of tenets are research, education, and innovation. They work very well with each other and train a lot of orthopedic surgeons, and from that they get a lot of research expertise, basically to create devices and improve patient care. So excellent model. It's been in practice for a while, and that's what we're trying to follow and develop here at IR. This is a really interesting paper. It's actually more interesting because I realized Dr. Murphy, I was looking back at this paper again, he's the one who authored it, but it's a study of inventiveness in interventional radiology. And so IR isn't really that far behind, but what you notice when you read this paper is that there's a few major inventors who are responsible for a majority of the patents. It's not all the way through, or basically it's not everyone in every single institution is creating sort of the same amount of patents. It's coming from a few main centers. And if you look at this social network map, it shows you that there's few centers that are associated with the main inventors, and what they come come up with in terms of the conclusions are that large academic interventional radiology departments really are intimately associated with inventiveness, and it's because there's a few key enablers for that. It's mentorship, motivation, and industry contacts. And what gets in the way of this is the lack of mentorship, lack of industry contacts, and the lack of legal advice. So that's what we're trying to form with the RFS. And so how are we going to do that? It's a three-phase process that we envision. So it's the webinar series is the first thing. It's going to be a way to educate the IR trainees and current IR practitioners in the device development process. Now, this is borrowed from a really good textbook. It's called the Stanford Biodesign process, and this is borrowed from that, and it divides it into three phases: identification, invention, and implementation, and subphases and different topics, which we're loosely trying to get speakers to hit all these topics. And so this is going to be phase number one. Phase number two is going to be applying that into developing a list of needs in IR today. So I like to call this the IR skunkworks, and if you're not familiar with what skunkworks is, it was a division of Lockheed Martin quite a number of years ago, 34 years ago, and they were formerly known as the Advanced Developments Project, and they were responsible for really major developments like the U-2 bomber, the F-22 Raptor, SR-71 Blackbird, and it's basically what they do is get the best and brightest in the field, get them together in a room, give them all the resources that they can, and they're basically tasked with very important sort of changing or disruptive technologies and projects. And so that's going to be sort of phase two, applying this education to creating a list of needs in a structured format. And we have ways to help you out with that. It's a needs prioritization matrix where you come out with a bunch of needs, and then there's different distribution of weights applied to different factors that should be taken into account. We gather that, and we'll have sort of industry experts help you along the way and rate your idea to help you select which ideas are the best. And so from that we go into phase three. What's that going to be? It's going to be academia-industry collaboration, sort of the AOSynthese model, and take that forward into transitioning benchtop ideas into bedside and patient care actual products. So that's sort of the whole overview and the goal with the IR Innovation Program and the webinar series, and where we are going to be going forward with this. So, you know, everyone talks about the pillars of IR. What is it? It's clinical, imaging, and procedural, and innovation is really the fourth pillar of IR. It's not only important for us to work on it, it's you know, as the American Medical Association statement said, it's really our ethical and professional responsibility towards interventional radiology. And so with that, I'm happy to introduce our panelists for today. Our first speaker is going to be Dr. Arepally. He did his medical training from Emory University and diagnostic radiology residency as well from Emory. Then he was a faculty at Johns Hopkins he did his IR fellowship at Johns Hopkins and stayed on as faculty for about 10 years. And now he's the section chief of interventional radiology at Piedmont Healthcare in Atlanta. He's got a lot of research awards, and he's mentored more trainees that I could keep track of when I was doing my research here. And then he's got a ton of patents in the field of MRI-compatible devices and embolic reflux prevention. And you know, the famous catheter is, I'm sure you've heard of, is the Surefire catheter. So we're happy to have Dr. Arepally with us here. Our next speaker is going to be Dr. Kieran Murphy. He did his medical school in the Royal College of Surgeons and diagnostic radiology in Albany Medical Center, followed by a neuroradiology fellowship at the University of Michigan and interventional neuroradiology fellowship at the University of Geneva. He was a section chief of neuroradiology or neurointerventional radiology at Johns Hopkins and chief of medical imaging currently at the University of Toronto. In doing my patent research on Dr. Murphy, I lost track, but these are just some of the patents he has in bone augmentation, imaging navigation, catheters and guide wires. There's a ton of other patents on there as well, so we're extremely happy to have Dr. Murphy here. He's also he received the Innovator of the Year award at SIR this year as well. So what we will do now is we can switch over the screen here to Dr. Arepally and begin the webinar. Let's see.
Hello, everybody. Can you hear me?
Yes.
We can.
Okay, I was just muted for a second. I just want to make sure. I want to thank everybody for inviting me to give this, I guess, the inaugural webinar on innovation, and I'm actually very honored to be doing this, and along with my former colleague Kieran Murphy, I'm excited about being able to help residents move forward in this kind of area. This first slide is actually a picture of the Wadi Rum Desert in Jordan I took, but to be honest, I put this slide at the beginning because this is sort of how I approached medical device development when I first started doing this. I don't have the vast experience that Kieran has. Obviously, he's done a lot more, and as you know, he's won the award. But what I'm going to share with you is my experience in how we developed Surefire Medical, and hopefully when you start your career or after training, you will approach medical device much different than this, where I felt like I really had to learn everything from scratch. And I want to share with you some of the lessons I learned and what worked, what didn't work, and how we really developed some of these devices. Can everyone see the next slide? I'm just going to ask that once just to make sure it's moving easily.
Yes, it is moving forward. Thank you.
Yeah, it looks just fine.
Okay, good. All right. So anyway, first, before we start, I want to give you like an overview, a little bit about what we're going to talk about. You know, especially in the industry, in the medical device industry, there's a word you'll hear called biotechnology. But really, biotechnology is split up into medical devices, diagnostic, and pharmaceuticals. Those are the three large areas of biotech industry that's out there. And obviously in this session and future sessions, most likely you'll be talking about medical devices, but of course there's other components such as diagnostic and pharmaceuticals. I'm going to be talking about medical devices from my experience. If you compare medical devices to pharmaceuticals, there's really two different lines here. And there's a big differentiation, and that's why you'll see the pharma world and the medical device world. If you look at it in terms of the pharma world, they're really long product cycles. It takes them a very long time to develop it, whereas with medical devices, they're much, much shorter. In terms of the pharmaceutical world, you have a lot of systemic effects. Because there's systemic effects, there's a lot more regulation involved in how these are approved. Whereas medical devices, the good thing is they're mostly localized effect, if you think about it, whether it's RF ablation, whether it's a catheter, a balloon, or a stent, or biopsy. It's all local, so you don't have that systemic effect that pharmaceutical does. So therefore, there's a little bit difference in their effect and also their approval. A lot of pharmaceutical is molecular-based, whereas medical devices is engineering-based. Although now these two areas are starting to slowly merge, it's not as clearly differentiated nowadays. Pharmaceuticals is more of a automated process. Once they figure out how to do this, it's sort of automated, manufactured, and then delivered and packaged. It's all fairly well automated, and you give the drug in a certain way. Whereas medical devices has a significant user interaction. It really depends on the interaction with the physician, the operator, or the surgeon. So that's the big difference. And then finally, the last two, I think, is really what's the interesting parts about medical devices. As you saw earlier, most of medical devices are physician inventors as opposed to pharmaceuticals, tend to be more lab-based research. And then the other thing is medical device, the really exciting part is that you can really do a lot with lower capital amount. Whereas pharmaceutical agencies, sometimes it can take up to $1 billion in research, R&D, to bring a drug to product to clinical trials, whereas medical devices, you can do it for a lot less. So I'm going to just walk you through some of these milestones. So this is a chart I have. The milestones on the bottom, and the value of a company is on the y-axis. So with pretty much any technology that you do or develop is going to go through these stages. And I'm talking about the stage that starts from a concept to prototyping to patent applications, animal testing, human trials, FDA approval, market introduction. Pretty much whatever you do, if you want, whether it's in any aspect of orthopedics to interventional radiology to even just minor procedures, they all will follow pretty much under these milestones. And if you start a company or associate with a company, these are the milestones you want to achieve. And as you achieve each one of these milestones, the value of what you are producing increases both on a technology standpoint, but of course also in a financial standpoint because you add more value to the device. So from our standpoint, the way we started Surefire was we didn't actually come up with, oh, I want to make an anti-reflux device. Actually, we actually followed the Stanford biodesign model. And the Stanford biodesign model, as probably most people will know, is a system or an algorithm to really find the need, then based on that need, invent something and implement it. So identify, invent, and implement. And so really, before we even started doing anything else, we wanted to first identify the need and build from there. So this is Jim Chomas. He's the man over there holding next to a microscope, a catheter. He is a co-founder with me of Surefire Medical. Jim is a PhD engineer, trained at UVA and Duke. So he had actually, when I left Hopkins in 2009, he approached me about wanting to start a company because we had a mutual friend that knew both of us left and were interested in starting a company. So one of the first things, he and I met and we said, all right, we want to start a company and really work on some device development, but we don't know what it is. So we really first wanted to identify what we want to do. So what we did was the immersion technique. Well, basically, Jim would fly around the country and go to cath labs around the country with a notebook with friends that I knew or he knew, and he just stayed in the cath lab and he just took notes and tried to identify clinical needs. I was the same way. As I was doing cases, as I was finding needs, I would sort of make a list. And then every 2 or 3 days, Jim and I would sort of reconnect and go over the phone or via Skype different needs and different identifications. And we really basically honed down our criteria, 3 things to really start our first company. One, we had to have a medical device in interventional radiology. Second is we wanted to have a really low-hanging fruit. That means we didn't want to go for a big, complex medical device. We wanted something that was easy, much simpler to sort of develop at the beginning. And then we wanted a straightforward regulatory pathway. We don't want any kind of molecular engineering involved. We want to make it very straightforward. And so those are our 3 criteria that we really used as we were trying to do the needs identification. So based on that, one of the things we decided to do was work on delivery. If you look at especially with embolization over the last 20 years, there's been a lot of sophisticated agents that have been developed. Here's a picture of a drug-eluting beads, but now we've got obviously glue, we've got detachable coils. So we've got a lot of bullets, basically, a lot of bullets we could give to treat patients. The imaging has also gotten really, really good. Now we have cone beam CT, we obviously have 3D imaging, and then so we have a lot more functional imaging. So the imaging has gotten really good. But the one thing we noticed was the delivery was the same. Everybody was using the exact same catheters they were using for the last 20, 25 years. And as we like to say, people were just using the same gun, but different bullets. They had much more advanced bullets, but didn't have any different kinds of guns. So we decided, let's make a better gun. Let's make something a little bit better in delivery. And so one of the things we did was, if you looked at the way we started it, was we basically looked at the complication rates, especially we started with radioembolization, where we deliver radioactive spheres in the hepatic artery for treatment of malignancy. And we noticed there was a pretty high incidence of GI symptoms. In this Cancer Journal article from 2010, about 25% GI symptoms. And then of those, you really have can have a pretty significant ulceration rate, really related, can be up to 20%, and this is all related to reflux. So one of the things we wanted to figure out is how can we better deliver agents so you can have improved targeting and less reflux. So with every startup, with every other things, you have a bar phase and you have a garage phase. Well, this is our bar phase. This is Quattro. This is a pizzeria in Atlanta where Jim and I talked and we sort of figured out what we wanted to do. We formed the company here. We basically wrote on napkin paper some of the ideas we had and really started to build what we wanted to do. And when we talk about a bar phase, the part where there's really nothing in the company other than you're hanging at a bar, this actually is really the fun part. I show you this picture here. This is a table with my book bag there, and basically Jim and I rented a room in an internet service provider for $300 a month. and Jim basically used that room, and I'd fly into Boulder, and we use this as an office to build, to do our bar phase. And what did we do? We did a lot of research. We just spent time reading, trying to really identify what is going on. And then we incorporated ourselves. We came up with a name, and then from there, we actually started writing a business plan. And from a business plan is probably one of the best exercises you can do if you're new to this, because that will really make you focus on what you want to develop. A business plan is like writing any other grant. There's certain elements you have to fill in, and once you write a business plan, that's sort of almost like your living constitution for the company you're working on, or the project you're working on. And I suggest everyone read business plans to sort of get an idea of how they're written, other ideas, but basically this is what we did at the beginning, and just sat down and wrote a business plan. Then we sort of went to the inventing portion. We started where we were doing brainstorming. And we did it several ways. You know, it was first me and Jim doing the brainstorming. Then we got a couple other engineers involved. Then we got an IP attorney. Then we got some regulatory people involved. And we all started brainstorming either face-to-face, and a couple of times we did it via Skype or other, go-to meetings like this. And we just really sat down and hashed out ideas and different approaches once we knew what the need was. We didn't try to hone ourselves in this one specific way to solve the problem. We just wanted to figure out all the different ways to approach this problem. And that's one of the things we can do. The other thing you have to do as you're doing this invent phase or the bar phase is you really need to start looking at patents and understanding and reading patents. So where do you go when you want to look at patents? Nowadays, there's just so many good choices of doing research. These are some of the online places you can look. I tend to use Google Patent Search most of the time, but the U.S. Patent Office is also very good. They have a lot of good information on their website. And in terms of the patent, you can do a whole series of lectures on patents, and so I'm really going to reduce the information down to really simple criteria. It's a utility patent, and you need to have 3 criteria for a patent to be successful. It must be something useful. It must be novel with respect to other patents. It must not be very obvious. These are very general statements, obviously, and it's much more sophisticated than that. But that's really the general criteria people use for approving a patent. Once you understand the patent standpoint, then you have to start thinking about the regulatory standpoint. Where does my device fit? Or if I'm developing something, what are the things I need to think about? Well, the FDA has really 3 categories for a device. So as you're doing the brainstorming, you got to figure out where you want your devices to go. Most likely you want your device to be in class 1 or 2. Now class 1 is hard, so this is a class 1. This is the one that has the least regulatory involvement. And this is the one that basically the best example is a Band-Aid. That's something the FDA is not really going to be hands-on with, and it's the easiest one, low-risk device. But obviously, pretty much any medical device that has any kind of invasiveness to it is not going to be a class 1. Class 2 is really what we were looking for. We wanted to do a class 2 device that's a little bit higher risk, is a little bit more complex than a class 1, as this slide shows you. The good thing about class 2 is you need a regulatory pathway that's really 510K, and I'll go over that a little bit. But 510K is basically a regulatory statute that allows you to get approval of devices if there's something already on the market. It's something similar to something already on the market. Finally, if you go to a class 3 device, then that's where you're really going to make yourself challenging. So as you're collaborating or brainstorming, you got to make sure you understand if this is going to be a class 3 device, because that's going to affect everything down the road, because class 3 devices are really devices that support or sustain human life. Very, very strict, rigid criteria. And the best example of a class 3 device is pacemakers, AICD. So these are things that have a very high level of regulation, and they're going to need pre-market approval. This is sort of how basically the general the devices sort of fall. You have class 1, class 2, and class 3. Pretty much everything falls in class 1 or 2 if possible because of the regulatory pathway. Like I said, most everything you will encounter is going to be a Class 2 device, and then Class 1 is very rare because it's such a small amount, and then Class 3, which is this 1%, is really the one that's going to require a lot more strict regulatory pathway, and it's a very small percentage of medical devices. So what is the 510K? It's basically what the 510K pathway says is that there is a product out there that's substantially equivalent to a device that's already marketed. So it means your device is it's very similar to a device that's already marketed, and that's your predicate device. So now you see the dilemma. You have a patent situation where you want to be as unique as possible, but from a regulatory standpoint, you want to be as close or similar to other things out there. So that's where the real the challenge is to, as you're doing your brainstorming, you want to be able to have something that gives you a straightforward regulatory pathway, but also from a patent standpoint, some very strong, unique features. So for the bar phase, we wrote we incorporated ourselves, we wrote the business plan, we actually identified the engineering team, identified the IP attorney and regulatory expert. Next is what we call the garage phase of the startup, which we then moved to, which is basically you find a team and you start building it. And literally, I kid you not, we did have a garage phase. This is Jim and I flying out to Sunnyvale. And ironically, the first engineer we worked with, this is Ron here, his name is Ron French. It's pretty great. I mean, a medical device catheter engineer, his name is Ron French. Anyway, this is Ron's garage, and he had we had worked with him in his garage to develop some of the early prototypes for our device. So then let's talk a little bit about the startup calculus of, now that you've started developing an idea, and now you want to build it and build a company on it. Well, here is what I call the startup calculus. What about the money involved? Where are you going to take the money, and how are you going to use it? Well, first of all, you're going to have an angel round. An angel round is really the beginning, where you have people, angels, good people that are going to give you money that allows you to use that money. And you're going to use that money to help get you to a patent application, hopefully, or at least get you a prototype. Then you're going to burn through that money, and that's the burn rate. And then after a while, you're going to then go to what most likely is Series A, which is the next round of funding. And hopefully the Series A will give you enough money to sort of then take you from all the way down to almost the FDA approval, and then this is the burn rate, and then hopefully Series B takes you from FDA approval, and then hopefully as you get to the market introduction, you're able to sell enough that you can then sustain the company and then build from there. And this is obviously a theoretical model, and this is the ideal situation, but we sort of followed this pretty closely. This is sort of how we ended up doing this, and our Series A was really used to go from patent applications to the human clinical trials. In terms of seed money, how are you going to raise everything else? Here's sort of what you can expect from the dilutional aspect. The seed money or angel money here can anywhere be from $50,000 to $1 million. You're going to be diluting 50 to 30%, and each level you can be more and more dilute. So just remember that's what sort of happens with any kind of fundraising you have. What about venture capitalists? We didn't go through venture capitalists, but obviously this is a very common situation where they come in and provide capital for ideas. Venture capitalists are really looking for an elegant solution to a major problem. So they want a unique device. They don't want another Me Too product. So that's what you want to look at. They want a market potential of $500 million. You'll know immediately what your market potential is when you write your business plan. And that's one of the things they want. Third is they want a team. They want to know who's the team, what is their track record, and will they come up with other ideas. And so they want to see what is the depth of the team. Big thing for the venture capital is their exit strategy. How do I get my money out? Typically they want to make 3 to 7x return. So they want to make a lot of money based on the investment. So that's one of the things they always look at. And then finally, the path successes. This is what really killed us from our standpoint starting with Surefire is we were new to the game. We did not have any experienced people as we started, and so a lot of venture capitalists passed on us, even though we made pitches, because we were unknown entities. But the more known entities you have, then you obviously will have more people being willing to give you money. What's the hierarchy of raising money?
Dr. Arepally?
Yes.
Sorry to interrupt you. We just had a question from somebody in the audience. Dave Rajaratnam is asking, what do you mean by dilution in the series funding on your previous slide, the slide just previous to this one?
Sure. So basically, when I say dilution, what happens is if you own, let's say you own 100% of the company, and you raise, let's just go straight to seed money, and you raise, $100,000, $500,000, $1 million, you could end up only owning 70% of the company at that seed round, because 30% is diluted, 30% goes to someone else. Then you go to Series A, then your initial 70% will then be dropped to 35% because it may be diluted by another 50%. So it's just percent ownership, equity ownership in the company when I refer to dilution. Sorry about not explaining that. All right, so now we go, is that okay in terms of?
Yeah, that's great. Thank you very much.
Oops, sorry. All right. In terms of hierarchy of raising money, here is sort of the general hierarchy. You start by doing money yourself, bootstrapping. You can do that. The other options are STIR, STTR grants. A lot of startups do this. It's a great way to get undiluted money. Basically, the government gives you money. It's free. No one else gets equity ownership in your company. You can do the family and friends route. Ask them to give money. You can have employees give money. Private placement are people that are individuals that have a net worth of greater than 1 million or over 200,000. They're willing to put money into startups. So these are all some of the ways you can start off at the beginning. And these are the mechanisms we actually chose with going. We actually started bootstrap ourselves, and then we built it this way. Then, of course, you can go into larger scale. We have angel fund, which is basically a group of private individuals that pool their money together. They're angel investors that can give money with larger amounts to individuals. Then you have venture capitalists. Then, of course, you have the ultimate exit, which is either an IPO, as we all hear about, where you raise money from public capital or people from investing, or then you also get bought out by a major corporation. So that's the hierarchy in general of how you want to do that. From our standpoint, we ended up having two individual investors, Norm Eldon and Karen Cassidy. Both are serial entrepreneur. Karen actually was the first woman venture capitalist in the United States. And that's Norm at the podium, who's an engineer. He's a PhD engineer himself. He's actually the Weldon Purdue School of Bioengineering is named after Norm Eldon's because that's where his alma mater is. But anyway, so Norm and Karen both invested in Surefire from the beginning from a private standpoint. So they've been not only a great ally for us in terms of investing, but have tremendous mentorship and support for us. So they have been along with us every step of the way for Jim and I in helping us build this. So then we go from that aspect, raise the money, you've got the money, but now you got to do some testing. This is where I really started feeling comfortable because I've done a lot of animal work at Hopkins. This is the first Surefire we use. As you can see, I kid you not, that is a kitchen sink over here. So basically, that's the Surefire catheter. This is our very first catheter. It required a 9-French sheath in the right. This is the renal artery. You couldn't even see it. You can see how big the device was, and then obviously we deployed it in here. But this is the first animal study, and then we did. This is the early days of cone beam CT, trying to figure out do we stop reflux or not. And we think we did. We couldn't tell. And then after time, we really got well. We got to really learn how to do this really well. And from this, we actually started publishing our animal data because it was so well, it looked so good. So that's the other fun thing about doing this is, you're still publishing and doing research and all the things I was doing in academics. Now we're doing it from a startup standpoint. So the capital allowed me to really start doing some of the animal research and publishing some of this data. Oops. And then this is the Surefire system. It's 3 French now. Now we're coming out with the 2.6 French system, but you can see how we've moved it along from how it looked over the sink to now having a much more sleeker look. from there, you start doing clinical trials, and it depends. The clinical trials really can occur after FDA approval or before FDA approval, because the 510K just needs, if you have a device that's already out there, you can get theoretical approval. From our standpoint, we actually did some clinical trials beforehand. There is a pathway available outside the U.S. From us, it was from Sydney, Australia. Surtex actually called us and said, would you be interested in doing this in humans? We're like, okay. We didn't really even, weren't planning on getting it ready, but they said, can you do it Memorial Day? And we're like, okay. And then so for the next 3 months, we worked really hard to get everything ready for human use. So basically on May 29th, I took a 3-day trip to Sydney, Australia to do the first human study. This is us in Sydney. This is Lawrence Bester, interventional radiologist in St. Vincent's Hospital in Sydney. This is David Kay, Chief Medical Officer for Global Surtex. And this is David Liu, who's basically Chief Medical Officer for Surtex North America. All of us, we've been, obviously this is me, and this is our first Surefire case doing, going down there and doing this. And then from there, you do the FDA approval. We submitted the data. We didn't really submit the clinical data from Australia because it was so close. But anyway, we went ahead and submitted all the animal testing data. We actually engaged the FDA before we approved it in what we call a pre-IDE meeting. We asked them what kind of data they're looking for. They told us, and we did the animal testing and submitted it. And really, on June 24th, it's funny, how do you know you get FDA clearance? You think that there'd be all these bells and whistles, but actually it's funny. One day someone was walking around and they saw a fax laying around this machine. They picked it up and this is the letter. You know, you don't really get a notice from the FDA about just a generic letter and a fax machine. And this was stamped June 24th, but the fax came on June 30th. I mean, like, this was a huge deal for us. I mean, our first application, and it was just sort of almost nonchalant how quickly it just sort of came, and we didn't even, you know, you'd think there'd be like more pomp and circumstance for it, but it wasn't. Anyway, this is the timeline, you know, from concept to market introduction. You know, we really did it over about a year and a half, if you look at it. We worked really hard, we worked very fast, and we had a lot of fun doing this. I still say, professionally, this is probably one of the best things I've done, both on a intellectual level, but also on a social level, as well as just an energy level. This is just a tremendous amount of fun, and the people I've gotten to meet along this pathway has been very special and unique. So I'll finish up with a couple last slides about, I always get asked, like, what can I do? I want to have an idea. I always tell first things is, you know, you need to do some reading. You know, it's, I realize there's a lot more, and these are some of the stuff I read. These are 4 good resources from my standpoint. Biodesign is a book by Yock et al. This is the actual textbook that's available at Amazon that really goes over the process of medical device innovation. The next book is called Venture Deal. It's a really good book about the legal aspects of startups. I like their subtitle, which is Be Smarter Than Your Lawyer and Venture Capitalist. Really good way of understanding what dilution is, how companies are structured, how you want to do term sheets and liquidity, preferences, all that stuff. The other journal that's really good is this magazine called Startup Magazine. It's really about medical devices, startup phase. Has a lot of good stuff. I look at this just to read what's going on. It's not necessarily even related to Surefire. It's just startup ideas in the medical device sector. This can be fairly expensive, you know, but there's always past issues around. I always learn a lot about what's going on from that. The other thing is this one is actually available on the web. If you Google this, The Entrepreneur's Guide to a Biotech Startup by Kolchinsky, it's actually very good. It talks a little bit more about, it's a biotech, so remember when we said biotech, it talks about pharmaceutical and medical devices. So that's the good thing about it, is it covers both aspects of the medical devices, diagnostics, as well as pharmaceuticals. So I always tell people, read. If you have an idea, read all these and read patents. Google patents, everything else. The other thing is I always tell people is find a partner. There's no way you'll be able to do this by yourself. It's never, if you look at even the greatest startups in the world, none of them have been one individual, whether it's Facebook, whether it's Apple or Microsoft. it's always 2 or 3 individuals. So it's never the lone person. So find a partner, find someone that's got complementary skills. I was fortunate to have Jim, who's an engineer, and I was obviously an MD, so it's nice to have a complementary skill set. The other thing you can do is if you really don't have time to really, I mean, the essence is time. That's the most important thing you need. If you don't have time for this, then the other option is to do what I would consider is find a local incubator, and you can, partner with them and where they'll do that. Oops, hold on a second. Whoops. And then in terms of incubators, is if you're in academic centers, there's all these places that are available, like Northwestern has NUCAS, Stanford has Biodesign, Hopkins has Bioengineering Innovation Design that I worked with when I was at Hopkins. But there are also stuff out there that's not academically oriented. These are our private incubators. This is GCMI in Atlanta, which basically all they do is do device development. You come with an idea. There's another one, actually, ironically, this is also in Atlanta, called Hatch Medical. And this is the other one in Atlanta called Atlantic Pediatric Device Consortium. So wherever you look, you'll find a local incubator, but they also, these incubators work globally now. So they take ideas from anywhere. So you can always reach out to them. I'm always very interested in this one because I think the Pediatric Consortium, there's a big need for pediatric devices. There's an area that we really need to help develop and foster. It's this area, and there's a big opportunity there. An example of success for them is someone just came out with a otoscope that plugs into an iPhone. So basically, you can take a photo with your iPhone of an eardrum and then send it to your pediatrician. So these are all like simple ideas, but very powerful. So I'll finish up with one quote my mentor, Paul Bottomley, shared with me. The quote is, "The reasonable man adapts himself to the world. The unreasonable one persists in trying to adapt the world to himself. And therefore all progress depends on the unreasonable man." So I really want everyone to sort of think about being not only unreasonable, but think out of the box to try to think of other ideas. And that's my last slide. Those are all the people I've worked with over the last few years. And that should be it. I'll hand it back to you guys.
Great. Thank you very much, Dr. Arepally.
Thank you so much. It
That
was
was
a
a great
fantastic
talk.
presentation, Dr. Arepally.
Go ahead, Raj.
What we'll do is we'll hold the questions for the end. We'll switch it over to Dr. Murphy, and then we'll do questions right after that. So let's share Dr. Murphy's screen, and then we'll get going with that. Thanks.
Okay, can you hear me?
Yes, we can. Thank you.
Great. Well, Aravind, I really enjoyed listening to you. I had the pleasure of working with Aravind for 10 years, from 1998 to 2008, and it was a great working relationship. We're both fortunate to have the same mentor, a man called Elias Serhouni, who was the chair of radiology, and I think he influenced many of us and gave us great advice. He sat me down one afternoon when I was having kind of a shitty day, and just told me how to start a company and about how to raise money and how to get the idea out of my head and onto a pathway that would result in it being used globally. And he gave me great advice. He did the same for Nick Fernano, and he did the same for Arvin and many other people. And this role of mentorship is critical in managing your intellectual property. It really can't be underestimated. So I'm Professor of Radiology. I'm Director of Clinical Faculty at our Research Institute, TECNA, which is dedicated to biomedical engineering and physician-driven invention. I spend 2 days a week of my time doing nothing but that and 3 days working clinically. I also have started a bunch of companies, 6, 2 in France, 4 in the US, actually 2 in Canada, so 8. And I have a private equity life. I'm a senior technology advisor to a private equity group called TVM that has about 1.8 billion under management. So what am I going to talk to you about? I'm just going to try and hit some topics that I think are important to you. You've got to document what you know. Doesn't matter what it is. You've got to write it down, sign it, and date it, and get it co-signed. This is co-signed by my son. It's an idea for an expandable needle through which you can do biopsies. and it shows the time and date and the sketches around the idea. Now this is not bulletproof, but it does document your thought, and it is important to do this. So I've gone through about 13 of these notebooks, and when I'm meeting with anyone from industry, I have one of these notebooks in front of me. I staple their cards to the page and the date on which we met, and I document what we talked about. I do this because twice I've been completely ripped off by Johnson & Johnson, and I promised I would never let this happen again. They did that in 1997 for the first time, then again in 1998, and I try deliberately to work with privately held companies. Since then, I try not to work with publicly traded companies. I like to work with people like W.E. Cook, who are very much sort of a family business. Some of the larger companies like Bard are like this too, but many of the bigger companies that are quarterly driven by, they're publicly traded quarterly driven companies, they're often staffed by like high school freshmen who know nothing about our field. Whereas you go to these small private companies, you'll find guys who've been there for 10, 15, 20, 30 years from the very beginning of SIR, and you can be safer in what you say to them. So then, at a certain point, you file a provisional patent around your idea. These are cheap, $150, $200 to do something like this. And as Aravind said, you go through the patent filing process. Remember, it's first to file. It's not first to invent. So you have to file before you speak at a meeting, before you present your abstract at SIR if you think it's something valuable. There are folks who will sit in the audience, they'll take photographs of your slides, and they will steal your idea. And if you're lucky, after 2 or 3 years, or sometimes 5 or 6 years, you'll be issued a patent. When you have your patent, you can get a nice plaque. This is one of my most valuable patents. It's the one on the Cook vertebroplasty system, and this stuff is used about 60,000 times a year worldwide now. And I'm proud of that because it's very useful clinically. It's simple, it's cheap, and it's 20% the cost of kyphoplasty, but equally effective. So this was my first experience in inventing devices. I began as a fellow in Geneva developing bone cements, developing needles, working on cadavers at nighttime. My boss there, Daniel Rufinok, told me to speak to Cook, and I did. And they were great to deal with, and we prototyped some needles, and I mixed barium that I'd sterilized in the oven in my kitchen with PMMA, and we made radiopaque bone cements. And then I filed a wall of patents around that idea. I filed probably 11 patents around that idea to protect it. So it's a dense fence of intellectual property that I then assigned to Cook, and Cook repaid me my patenting expenses. So I got my money back in a royalty. And then they developed the market. And it was a very mutually beneficial relationship that's gone on now for 15 years. Sometimes the thing you patent or you work on, well, let me back up because it's been a long day and I'm a bit tired. Physicians are the best inventors. We invent what is needed. We invent things when we agree to do difficult things. When we agree to do a difficult case and we lie awake at night figuring out what the hell we're going to do the next day, that's when we're inventive. And sometimes we have a plan A and a plan B and a plan C, and sometimes we use all our plans. And sometimes we don't need to because everything goes beautifully. But there have been a couple of occasions in which I was down to plan C, and it was fortunate that I'd been up to 2, 3 in the morning ruminating on all the dark and possibly negative things that could happen in a procedure. I think most of the good ideas come from that situation where you agree to do something challenging. And when you're at the hospital of last resort, the folks who say, oh no, I don't do that, or that hasn't been shown to work, they're not being very inventive. It's the spirit of interventional neuroradiology, interventional radiology, that we say yes, and that we're the physicians of last resort. What you're looking at here are Just a simple thing. When you do a bilateral cement injection for vertebroplasty, when you inject the second set of cement, you can't tell where it's going until it passes beyond the boundaries of the first set of cement. So I double the density of the second set of cement. So you can see it against the pre-existing density. So this cement here would be twice as dense as this. And then when it starts to come back beyond the boundaries posteriorly of the first injectable load, then you can identify it quickly. So it's a simple thing to do, but it means you have two different types of PMMA. One perhaps with a density of 30% barium, the other of 50 or 60% barium. So you build this wall of patents. Now, I spend a lot on patents. I put most of my royalty income into filing more patents, with the expectation that this will work out for my best interests. But when you believe in something, that's, I think, the only way to behave. And there are a couple of drawbacks to being an inventor or to be believing in something that other people don't believe in. Your idea has to align with pre-existing belief systems. It's called techno-anthropology. So what we believe in today is probably in some way intellectually contiguous with what we believed 5 years ago. Now, if you come up with an idea that is out of step with that, that is quantumly different, that is like a Darwinian leap, it takes longer for people to adopt that idea and to assimilate it into their clinical practice. You will have a small group of people who will adopt it initially, often Australians, like Ken Thompson, because they're all half mad down there anyway. And you get the early adopters. But to get the middle bulk of people, which is the bulk of the market, it will take 5 or 6 years. One of the reasons kyphoplasty was so successful was they talked about correcting the vertebral deformity. And this aligned very, very well with the orthopedic belief system of orthopedic straight child. Whereas vertebroplasty aligned with what radiologists believe in, which is that we do embolizations. So this techno-anthropology element is actually very important in how difficult what you're trying to develop will be to get adopted. Now, I'm grateful to the first speaker for discussing my paper. This was a huge amount of work. I had great medical students working with me this summer. We cross-referenced the membership of the SIR with patent filings at the USPTO and the European Patent and Trademark Office. And we were able to show this massive growth of patent filings. And then we were able to break it down further, showing that many SIR members had 1 patent, some had 2 patents. And then a smaller and smaller number had larger and larger numbers of patents. And some people had huge numbers of patents. So this is one guy in Texas who had patented everything from wheelbarrows that pushed snow, called the snow barrel, to ways of tying your shoes, as well as ultrasound devices. So this inventiveness is clearly focused in the hands of a small number of people. In fact, 447 people at the SIR held 2,500 patents. So about 5% of the SIR membership, 5–6%, were responsible for the bulk of the IP around the entire field that is SIR and the multi-billion-dollar industries associated with it. Now, that's just patent filings. It doesn't cover procedural inventiveness. It doesn't cover the number of ideas that these people gave away for free to industry who then went and made something. Inventive people tend to have inventions around many other areas. Einstein has 33 patents, not related to physics. This is his patent on a refrigerator with the great physicist Slizard, who was one of the key people in the development of uranium-238 atomic weapons. Inventive people are just generally inventive, just like funny people are funny. And in fact, inventive people tend to be funny, and there may be something related in how their brains work. And Americans, in particular, are inventive by comparison to other countries. Canada does very badly here with only 16 patents, by comparison to the 1,617 patents and 938 applications. Sorry, 1,607 patents or patent applications from the U.S. So there's something fundamental to the U.S. character that indicates a strength in this area. So why would Canada suffer? Well, in this area, Canada has an innovation deficit. It's quite curious. It's really a problem, in fact, and the government is throwing money at this problem. And it seems to come from a certain level of comfort and a lack of physical and environmental challenges. So when you break this down, you can see areas that are under active at the moment because they've probably passed through a phase of incandescence. So around 1998 to 2004, around vertebroplasty, kyphoplasty, a ton of patent filings. In the stent world, you had Julio Palmaz and Grunzig, and then you had drug-eluting balloons, and since then things have tapered off. In MRI, not really that many patents because they're dominated by the big manufacturers. In embolization, you can see peaks for PVA and drug-eluting polymers, and then Y90, etc., and then tapering off. So each field will go through a period of tremendous incandescence and then quieten down. And because of the time lag between patenting and clinical approval, you can actually predict what we're going to be doing in the future. If I look at this, for example, so endographs ablation, obesity, clearly a really interesting growth of activity in the area of obesity-based interventions for people who are obese. Now this may be what Cliff Weiss is doing with embolization, or it might be maybe Aravind has something in this area. Maybe these are meshes implanted in the stomach, but an enormous amount of activity in the area of obesity. So that's this curve again in summary. Now the first speaker also spoke about those things that enable personal motivation, mentorship, institutional support, contacts, legal support, financial support. This issue of the social network is important. This is really about mentorship and culture. Culture of innovation is an institutional thing, and it took me a long time to understand that buildings have cultures in them that are a critical thing. The Hopkins culture, the UPenn culture, the Stanford, the University of Virginia culture, Emory. Each building has a culture that is derived from the founders of that institution. It goes all the way back. And if you don't have the right culture, you can't build it. And if you don't align with that culture, you should probably move. There are individual people at each institution that are critical hubs in these groups of inventiveness. If you see, say, Mahmoud Razavi, right here in the middle, a critical, quietly spoken, really quite brilliant, serially entrepreneurial individual. Gilles Soulez in Montreal. Bill Culp in Little Rock, Arkansas. Just a wonderful guy. Bob Vogelsang in Chicago. And these individuals are critical for mentorship, and so they play a key role in influencing other people. And I could trace in a time-elapsed model, say a person who had worked with Mahmoud would move to somewhere else, and then around that person would evolve a culture of device development. So we're like elements in the periodic table. I like this analogy very much. Primo Levi, the great Italian writer, used it first. And he assigned to people in a concentration camp different elements of the periodic table as evidence of their character, what kind of people they were. So you'll have some people that are like carbon, and they will form social networks. There are some that are like helium or a noble gas that float around and aren't that interactive. You'll have some that are more energy delivering, like iodine. And you'll have some that are like, sulfur or phosphorus, and they will be toxic to be around. And each department has an array of those people. The names may change, but their characteristics are the same. There are some individuals in our history that have just been stunningly brilliant. And Nikola Tesla, I'm sure Arvind will agree, is probably the most brilliant of all time. Not a normal person, but normal is not that healthy. in this regard. You want the abnormal. The great leaders in our field have always been eccentric. And that's the only problem I have with the Stanford Biodesign program, is that these people would not fit into that program. The Winston Churchills, who was a great inventor, would not fit in the Stanford Biodesign program. That's great for process analysis of a clinical problem like decreasing blood loss in the OR, but it's not going to give us the RF coil. It's not going to give us, you know, the first angioplasty balloon. This is Grunzig, who invented the first angioplasty balloon, and he as a personality is fascinating. He actually flew his airplane into a mountain while refusing to believe air traffic control that he was going to fly his airplane into a mountain. It gives you a sense of the strength of his character. Sorry for the bad photograph, but this is Julio Palmas, a very different person, inventor of the coronary stent, the Palmas-Schatz stent, a device which his institution told him was worthless, and they took the rights to a laryngoscope for intubating mice instead. Now, it shows you that institutions are sort of often the wrong place to analyze ideas. And one of the hardest things you'll do will be get your idea out of your institution. It took me 9 months recently to get my idea out of the hands of my institution so I could start a company around it. So Arvind and I benefited from the guidance of this person, Elias Serhouni, who was a first-generation Algerian immigrant to North America, a gifted leader, went on to run the NIH and now runs Santa Fe Winthrop Research. Really quite a brilliant individual, very generous with his mind. I want to just tell you things you may not know. You know, physicians have traditionally been great writers, poets, and artists as well. We have a tradition of being politicians. We excel in other fields of science. And so over the last summer, I had a bunch of students with me, and we made lists of physicians who are poets, artists, scientists, going back for the last 250 years. And we looked at, say, physicians as criminals. And interestingly, we found that 80% of the murderers who were physicians were surgeons, which is quite odd. Physicians as athletes. And so we found, I wanted to find out if there was a fall-off in engagement of physicians in other areas as we became busier and busier, and whether technology perhaps became the outlet for our other energies. And in fact, I found there was no decrease in the level of artistic endeavor by physicians or political endeavor of physicians at all, while there was this growth in technology. When you combine that information with major milestones in medical innovation, what was very curious was to overlap the artistic output and the major innovations of physicians. And what I found was that these peak in the years after major conflicts. So after the First World War, after the Second World War, after the Korean War, after the Vietnam War, physicians become more engaged in writing, in politics, in artistic endeavors, and in medical device invention. So this engagement of physicians as good citizens occurs in the time after major conflict. And I think this is sociologically quite interesting. In terms of women innovation, only 5.5% of patents are held by women. This has increased in recent years. Now 18% of patents filed since 2000 include women as inventors. There's a long tradition of brilliant women inventors. Ada Byron and William Babbage invented the first computer in 1835. Émile de Châtelet, the girlfriend of Voltaire, translated Newton into French and was a great inventor in her own right. So hopefully this is something that will be balanced in the future. Those graphs I showed you earlier demonstrate, as we know, that war is the greatest source of innovation. And sadly, but truly, it is where many of the new technologies will appear. Nitinol, as you know, comes from the Navy development of the metal for submarines. Perspex in the windows of Aircraft in the Second World War became the source of the first lens implants, and then Charnley used the same material for PMMA. Inventions arrive in clusters. The conceptual and technological pieces come together to make a certain idea imaginable. Around the world at the same time, there is often a sort of Jungian synchronicity where multiple groups will be working based on the same fundamental assumptions about how a problem can be solved. And this goes back a long way. Newton and Leibniz fought over who invented calculus first. The Germans, the British, the Americans, and the Japanese had atomic bomb programs. This is why it's important, this concept of synchronicity, it's important to file early. I'm going to skip that. Okay, I'm going to skip this. Oh, don't be trapped by dogma. Yes, don't be trapped by the results of other people's thinking. Okay, I'm going to skip all this stuff. I want to give you specific ideas around things. So I think there are things that we do that are stupid. And we have to look at everything that we do and analyze it and think, is this reasonable? One day, IV chemotherapy, we will think it's crazy. Whole spine radiation, we will think it's crazy. When we place a stent in a coronary artery, as I had done recently, cardiologists know it will fail unless the patient is put on aspirin and a beta blocker because it's a systemic disease. But when we do bone interventions, we don't approach it in that way. But we know that if somebody gets put on PTH, parathyroid hormone, Forteo, we can grow new bone. So I've spent about 7 years working on the concomitant use of calcium phosphate cements and subcutaneous parathyroid hormone. And so I've been able to grow wonderful thick bone by using absorbable, inductive, conductive bone cements that are injectable and PTH at the same time. And so this has been a very exciting project, actually, and it works to assist in the development of fusion. These are patents that I have issued. And the patents involve delivery systems, because why do we use lure locks? Lure locks are flow limiting areas, right? So these are non-lure lock syringes. The connection is on the outside of the syringe, not the inside. So non-lure lock syringes delivering cement, and delivering the cement using vibration, not hydraulic pressure. So a ball bearing in a raceway around the handle of the needle vibrates, and then the cement is injected under vibrational pressure. And this is integrated into the handle of the needle here. So low pressure injection of the cement, systemic delivery of PTH, and then you protect this idea with a wall of patents. So there are now 32 of 40 patents issued protecting this idea. Okay, does it work? Yes, thank God it works. This is a cadaveric bone specimen, traditional bone cement injection, not very good distribution. This is with vibration. Nice dispersion of cement throughout the vertebral body. Where did I get this idea from? I was watching a cement truck delivering cement to a building, and I realized that the delivery system was vibrating. That's what I copied. So you patent the heck out of something. This is my ozone device. I was at a meeting in Italy, and they presented 50 cases of patients having their disc herniation treated with ozone, O3. So basically on the flight on the way home, I doodled this thing. The batteries are here, and the software is here, and essentially you aspirate up oxygen, you turn on the device, and you get an electrical discharge that is like lightning within the barrel, and you get 2% ozone, 98% oxygen. This is what it looks like when you do it to a rubber glove. This is just O3. So O3 is remarkably aggressive, and it shrinks the nucleus pulposus. This is the device. That's just O3 being injected onto a regular rubber glove. So we've just finished a 40-patient clinical trial of that. What else have I done? This is my radiofrequency ablation device, and this is being licensed by Benvenue. We took their device and I turned it into an RF generator. It's a nitinol coil. We can deploy it in vertebral bodies, and we're developing radiation therapy type software planning systems to look at the energy distribution so that I know exactly where the energy is being delivered, and we can plan it like XRT and SBIRT or any of the other radiation therapy type approaches. With my friends in the JAN, we've taken accelerometers from the surface of submarines. These are accelerometers, listening devices, all along the surface of the submarine, and we built a headset that feels the vibration of cerebral pulsation with systole, diastole. And so we can monitor brain pulsation with inflow of blood and relaxation of the brain. And it's slightly asymmetric. Left side gets the blood first, right side gets the blood first, left second, and it's the pump that drives CSF flow. So the brain is a pump that drives CSF flow. And this is a normal tracing. So this device has now been shown to be very good at detecting stroke, which is asymmetric, but it's also good at detecting concussion as a loss of autoregulation of cerebral blood flow. Radiation. So, terrible photograph, but these are a bunch of interventional cardiologists, and none of them have hair on the outside of their left leg because they have radiation dermatitis from too much exposure. And this is related to radiation impinging upon water molecules, breaking the water molecule, releasing free radicals, which then attack DNA, and then the DNA binds with oxygen, so you get an oxidative injury to your DNA. And to counter this, we've developed a technique of using antioxidants. So where did this idea come from? So my mother-in-law came home from our cancer hospital, Princess Margaret, with a list of medications not to take prior to her radiation therapy for her breast cancer. So I looked at that list and I thought, geez, if antioxidants are strong enough to impair, centigrade and gray of radiation, surely they can protect patients against millisieverts of a diagnostic head CT or a mammogram. So over about 4 years of doing experiments on myself, we've developed a cocktail of 5 antioxidants that can successfully block DNA damage. And here is our first prospective randomized study. I presented this at SIR. These are 5 patients who got technetium MDP bone scans. The blue is pre-radiation, the red is post-radiation, and they all had more breaks after. This is the group of 5 patients we pre-medicated with 5 simple antioxidants, and you see that we can block nearly 90% of the DNA damage. So this we're going to develop into 2 pills, one fat-soluble, one water-soluble, that will be taken 1 hour prior to every diagnostic study, every screening study, every interventional study. I failed to do things, and there are some brilliant things that I really believed in that have failed, and it's important to discuss the things that have failed. The best thing that I ever did was nitric oxide coating on catheters to prevent vasospasm. This is an ill patient with a large ruptured PCOM aneurysm and spasm right through the ICA, and I coiled it. And you see the spasm around my catheter in the cervical carotid. This is a catheter down here coated with nitric oxide eluting polymer, spasm in the feeding vessel, and then just placing the nitric oxide catheter in the vessel, and the downstream elution of the nitric oxide resolves the spasm. Here's another example. Severe spasm pre-nitric oxide, then just having the catheter in the limb of that vessel, and all the spasm goes away over 30 seconds. This would decrease the risk of dissection, it would of traumatic catheter-induced dissection or thromboembolic events, and I couldn't get anyone to do this. and this failed. And I think, to be honest, it was really quite brilliant. Are there other things we do that are not great? Well, I broke a rib last winter, and I realized that rib fractures are incredibly painful and also potentially fatal. In fact, if you look at people with flail segments, those who get surgically fixed spend an average of 9 days in the ICU, and those with flail segments who do not get surgically fixed spend an average of 21 days in ICU. So I've developed a balloon-mounted system for fixing rib fractures from an intramedullary approach, navigating in the long axis of the rib. So a lot of what I come across is by personally experiencing things and thinking, there's got to be a better solution than this. This is the current orthopedic approach. We can do so much better than this. So your focus is really, really important. Tenacity and resilience are key, and mentorship is key in this process. One other thing, I think isolation is really important too. I'm sure Aravind may feel the same. The only time I get left alone is on an airplane, or if I'm awake late at night for some reason. But you need time to assimilate and integrate ideas and percolate them and really analyze them deeply. A good social network is really, really important. I work with the same 10 or 15 people all the time. And a lot of them actually are in Utah, where there's a great hub of biotechnology. I read, as Aravind was saying, I read all the time. We are what we read. I'd like to advise you to read some books. The Medical Device R&D Handbook by Theodore Kuchlik, and Aravind talked about the Stanford Biodesign book. The second edition just came out and has very good web resources with it. So conclusion, question dogma. The Irish are always very good at that. We're argumentative. Solitude or a nap. You must be a content specialist, but be aware of what's being done in an adjacent field. Watch out for techno-anthropology that can trip you up. Follow the thread when you have an intuitive perception of an alternative pathway. You may have seen the movie Divergent. Inventors are fundamentally divergent, but we're constructively anarchic. Engage and say yes to difficult things and give a damn. And remember, we're not just here to do the job, we are here to change how the job is done. So I hope that was useful. Thank you.
Thank you, Dr. Murphy. That was a fabulous talk.
Anyone there?
Yes, can you hear us?
Great.
Dr. Murphy, I'd just like to start with a question that I've had. One question that always comes up when discussing patent is how they interact in kind of an academic setting where you know, ownership by the academic institution is often may kind of interfere with some of that, and I think you briefly mentioned this in your talk as well. I'm just kind of curious, has much of your patent design occurred while at an academic institution, or how do you go about being innovative kind of on the side or setting up startup companies?
So I've only ever worked in academic institutions, and you know, I was fortunate when you're a fellow or a resident, you have a great deal more freedom than you do when you're staff, because you're often not restricted in who owns what you think when you're a resident or a fellow. So at Hopkins, it was a very fair system, and I would disclose to a woman called, I think it was Julie Gottlieb, I'd tell her what I was doing, and you know, I did a lot of work at home. I never had a lab. I didn't use any hospital resources. I didn't use any bench space to do most of it in my head, and you know, I didn't really use animal labs. But if I did, the rules are very black and white. If you're using hospital resources, the hospital owns a piece of what you do. Now, since I moved to Toronto, I have to say I struggle with this. But my radiology group believes that they own at least 25% of everything I invent, and the hospital believes that they own 50% of what I invent. So I wind up giving 50% to the hospital right off the bat, and of the remainder 25% to my radiology colleagues who are golfers. They play golf, you know, and that really bugs me. But if you want to understand why Canada does so badly in the World IP Stakes, it's stuff like this, because there's no incentive, you know. So my role here is to mentor and develop people's careers. So we filed about 15 patents in the last 5 years. We've started a bunch of companies. And I play by the rules because I have to be a role model, but it's not a conducive environment to invention.
Thank you.
Arvind, what do you do?
Just unmute Dr. Arepally.
Okay, great. Great lecture, Kieran. I have patents at Hopkins that have gone through the process, but since I left Hopkins, I'm sort of independent. When I left, one of the things I did when I joined my group was very explicit in the contract about patents and everything else, and I was in a private group. They really had no interest in patents. They were more concerned about, obviously, clinical work. So I made a very specific carved-out provision relating to patents, intellectual property, and all that stuff. So that's different for me because of the private sector, and in a way that's beneficial for me because I've already had the academic experience and been able to parlay that into what I do now. So I'm fortunate from that standpoint. But I think it's a very important question you have. Wherever you go, you have to be very clear, understanding of the proprietary risks of who owns it, and be very clear before you start who owns what.
Great. We have another question from the audience there. I'm just going to unmute him. It's David Rajaratnam. Go ahead, man.
Hi, Dr. Arepally and Dr. Murphy. I just want to say thanks for the great talks. It makes me feel like you guys were my mentors for the last 45 minutes, so that's pretty incredible. But I'm at a kind of small to moderate size university level in a university, and I was just wondering, when you have found a need and developed a concept for a device, how would you find an engineering or computer science expert to help you develop that concept before you actually get to the prototype phase even?
You want to go first, Kieran, or?
Well, who's around you? What hospital are you at?
Loma Linda University.
Okay, so there's lots of good people there, I'm sure, right? Do you have a biomedical engineering department?
We have a small one.
Yeah, and is there anyone in your department who's invented something or has any experience in this area?
Not really. That's one of the problems I've been having.
All right. And what about other departments like orthopedics or cardiology or some other department?
Actually, I actually haven't looked down that avenue. That's a really good point.
And what about, do you have a tech transfer office there?
I'm not familiar with that. What's that?
Okay. So there'd be an office of, you know, they might have the name Innovation or Technology Transfer or something, you know. And the other thing is just, you know, Arvind gave you some names of groups of people who could facilitate this. I work with the same group of people in Utah all the time, and they're called ZIEM, Z-I-E-M, and they can make anything. Like my last idea of the needles, they made for $200-300, and they were terrific, you know.
Wow.
And they FedExed them to me at SIR. And then instead of sketching something, I was able to show something to people. And with 3D printing, getting your idea turned into something that can be held is so much easier and so much cheaper than it used to be.
Mm-hmm.
I was going to suggest, I think one of the things as Kieran also alluded to, is trying to find a culture in your local area of innovation. And how you find that culture is not only you'll see local lectures, obviously, but also find some angel groups. There's, especially here in California, there's a lot of angel groups that meet. And you can sort of reach out networking through them. They usually have some kind of networking process in place where a lot of engineers get together. That's one. Also, at any major city, you're going to have medical device experts, and you can even do via Google patent search, find out what kind of engineers or where they are, that kind of stuff. So there's all kinds of information available out there. You can even just go to engineering schools, local engineering schools, and find out, what kind of seminars are going on, and actually introduce yourself to some of the mechanical engineering or chemical engineering or electrical engineering people, and see if they can give you names of people that are working in, specifically biomedical.
Great. Thank you very much. That definitely puts me on that track on where to get started. Thanks a lot.
Great. So we have another question here from Evans Hytheus, and this is for, I guess, Dr. Murphy and Dr. Arepally, both of them. It's what's your experience in international patent law, and how do you deal with an idea born and bred in the U.S. and protecting it abroad? And do you have the same problems in Canada, Dr. Murphy?
Well, you have to file after a certain number of months internationally if you want international protection. The whole trick really is to get rid of your patent within about 30 months, otherwise it gets stratospherically expensive. When you start out to do individual regional filings, or even if you get your patent issued and have to select what other countries you're going to go to, you can easily get into the $50,000, $60,000 range per patent or more. And so none of us can afford those type of expenses. So you want to have a partner before you get to that stage. And this requires focus. And it also requires, say, selecting what countries you're going to file in. Like Germany is a market where they pay their bills. Italy doesn't. Greece doesn't. And, so you don't file there. China is actually worth filing in. Australia is worth filing in. Spain wouldn't be. And so you make ruthless decisions about where you file based on the market. The best market in Europe is Germany. France is probably second. So, you just have to be very cautious and watch your dollars. You might want to talk to your patent attorney and say, I will pay you X for this whole process. Don't let them bill you by the hour. Work out a number that you will pay for that bundle of work, and it'll be a lump sum. Otherwise, for every NDA that you sign, every term sheet that you have them review, it'll be another $1,200, So get them to decide per piece of work how much they're going to charge you and negotiate that with them aggressively, and don't just wait for that bill to show up.
Yeah, I agree, and I think I would first, especially as you're beginning this process, I would stick with learning about the U.S. patents and getting that done, because that's the most powerful in general. And then obviously it takes a lot of money to go to every country and do that, so I wouldn't worry too much about that right now. You can learn about it, but I would try to stay concentrated in the U.S.
So there's another question kind of in terms of forming the idea. I know like a lot of people believe in instead of like a device-first approach, like where you have an idea for a device and you try to force it into a market, what would be a recommendation in terms of coming up with an idea in terms of a market-first approach and a patent-first approach where you do the market research and figure out, where is the actual need, and how would you go about that? Like, where do you look for? And Dr. Arepally gave the example of looking at the complications for embolization and going with that, or do you look at CDC data, or how do you compile that list of like the greatest needs in IR right now and what to target?
I think one of the strengths we have is doing clinical cases, and as you're doing clinical, you encounter them on a daily basis, and and there are things that you overlook. And one of the strengths and weaknesses of interventionalists in general is we tend to compensate for our deficiencies by coming up with our own solutions, but sometimes they're not elegant, and then we get into these habits of doing it the same way. So sometimes you just sort of step back and say, what are these habits that we're doing, and can we do it better? Can we improve ourselves? And just sort of if you watch as a neutral observer just some cases, watch other attendings doing cases, other people. People even talk about videotaping themselves to watch, and just slowly watch and just sort of immerse yourself and figure out what it is. And as Kieran said, have a notebook, write down ideas, write down. They're not all going to work, obviously, but it's the process of going through it, the exercise of thinking critically, and the exercise of trying to think it differently. I mean, you can't look at the same problem with the hammer and nail approach. You can't be using the same hammer. He said, there's got to be a different way of doing this. Can we achieve it? Can we effectively have a better effective result? So just the exercise, I think, is very important. Just going through that exercise in your mind and doing it on a few things. Other example is just pay really close attention to simple procedure. Really close. I mean, detail, every single detail, and absorb it and learn from there. And that's some of the things I would recommend for people beginning in this area to try to figure out what are some of the needs. That's sort of more on the individual side. On the larger scale, obviously I think reading is very helpful, seeing other specialties and seeing challenges they're doing and how you can incorporate some of the novel ideas in other areas and bring them to what you're doing. That's the other aspect of it.
I agree with Aravind. I would add that you know, your own personal experience is important. I had a myocardial infarct in January, and I discovered that getting an IV is really painful. Having blood drawn all the time is really painful. And, you know, so it occurred to me with a nurse, actually, that, you know, why not mix lidocaine in the alcohol swab? So when you swab the skin, you numb up the skin at the same time as disinfect it. Or have them in two separate little swabs that you open simultaneously, so you apply both at the same time. These kind of kind, gentle things would be welcomed by patients globally, and that's a business opportunity. Or EKG pads, you know, they tear off your skin when you try and remove them, EKG leads. So this is a badly designed device. So we need to come up with a better design that doesn't hurt the patient when you try and remove them. There are lots of little things that we can do like this. There are whole areas that are done appallingly badly, like ventricular peritoneal shunt catheters, who fail 100% of the time. And when we put them in and take them out, we pull brain out when we do that. And these kids end up really hurt over time by these repeated tract hemorrhages, infections, repeated shunt failures. But it's an area that none of the technologies around catheters and devices that we use in the body have been applied to yet. This is a different silo. So I think there's an inexhaustible array of badly done things that we could look at.
I would say one other point I'd like to make, and you know, we talk about getting ideas from other places. I'm a big fan of that, but one area that's sort of even more interesting is it doesn't even have to be medicine. It can be some kind of hobbies you're doing. And one of the things about Surefire is I'm a sailor, and so I sort of know about how wind direction affects directionality of flow and how sails function, and that's sort of how we sort of came up with Surefire, is like having a sail on a catheter. And so you can use even some of your fun personal hobbies to sort of develop some of this stuff. So there's no boundaries in where you get ideas from. So just remember that.
I totally agree with you, Aravind. You know, I needed to make some things to try and hold needles, so I went to Williams in Sonoma, and I was looking at all the stuff that picks up pasta and picks up things in boiling water, you know, to try and find better gripping tools. Or you start trying to think of how am I going to change my gloves to grip a microcatheter better, or why are my gloves smooth? And how would I change the fingertips of my gloves? So this is a very good principle that you bring up. Darwin had a great interest in pigeons, and it was that knowledge of pigeons and raising parakeets that led him to begin to think about quantum leaps in evolution. So our hobbies do potentially really supportively infiltrate into our device creativity.
Great, thanks so much. There's a quick question about the books that you recommended, Dr. Murphy. Could you go over them real quick for one of the audience
Same
members?
as Aravind, the biodesign book by Yock, Y-O-C-K. Yock invented the monorail coronary stent. and I think he was involved in IVUS, intravascular ultrasound, as well. And he runs the biodesign program at Stanford. And the other book is more of an engineering book. It talks about how to make a catheter, etc. It's the Medical Device R&D Handbook by Theodore Kuchlik, Kucklick. And this is more of a, you know, how do you make a catheter, how do you code a catheter type thing. It's important to read that stuff just so you can speak the language.
Fantastic. If we don't have much more questions there, I know we're over time. We're holding people back here. I'd just like to thank again Dr. Arepally and Dr. Murphy for spending time with us and going through their experiences and all that advice. So just to sum up, this is the first IRIP, the IR Innovation Program webinar. We're going to be following up in a few weeks with Dr. Sanjay Srivastava. He's going to be the global marketing director of, or he is the global marketing director of the vascular division of Medtronic. He's going to be talking to us about needs creation and device creation and concept generation. And then to follow that, we'll have Mr. Walter Ryan, who's the VP of Business Development at Cook, who's going to be talking to us about how to create a collaboration with the industry and how exactly you go about that. So really excited about those. We'll keep you posted on when exactly they are going to be scheduled. And just to sum up some points from today's webinar that both of our speakers brought up, you know, partner up with someone. You can't do this by yourself. It's extremely hard. Document everything. Have a book with you that you write really great points on, or all your inventive ideas on. Find a great mentor, and that's what we're here to help out with as well through the RFS. Cultivate a good social network, and that's part of the IRIP mission statement. There is, the big centers are not the only ones that are going to be responsible in the future for all these medical device innovations. It should be everyone, and that's what we have to do, kind of create that inventive culture. Read a lot, be attentive to the world around you, like hobbies, and finally question dogma. So with that, thanks so much for everyone joining up, and we'll see you in a few weeks for the next webinar. Thanks a lot.
Thank you. Hey, Aravind, thank you for doing this. It was a pleasure.
Thank you. Enjoyed it, everybody.
Cheers.